Good day, students. Your active presence here is not just about attending a lecture on membrane permeability and transport mechanisms, but about participating in a shared exploration of knowledge. Let's make this session a dynamic exchange where listening, questioning and reflecting will turn complex concepts into lasting understanding. I invite you to stay attentive and curious as every detail we explore will sharpen your scientific insight following the learning objectives. First one is to analyze the principles of membrane permeability by evaluating structural models. Next is to differentiate between passive and active transport mechanisms. Next will be to examine the molecular structure, mechanisms and biological roles of ion pumps, to evaluate proton pumps and ion transporters, to evaluate the role of calcium signaling and calmodulin regulation in cellular processes. Next would be the evaluation of techniques in transport physiology, and lastly to integrate the transport mechanism in disease contexts. Dear students, we will now first start with the introduction to membrane permeability. The plasma membrane acts as a selective barrier separating the cells from surroundings. From Gotter and Grendel's bilayer model of 1925 to Singer and Nicholson's fluid mosaic model of 1972, membranes are dynamic, asymmetric structures. Membrane permeability enables nutrient uptake, waste removal and neuronal signaling. It maintains homeostasis by regulating entry and exit of substances. Composed of phospholipids, proteins, cholesterol and carbohydrates, membranes allow non-polar molecules to diffuse while restricting polar ones. Proteins govern transport specificity and functions include maintaining gradients, signaling and metabolism. First, we will discuss about the factors influencing membrane permeability. First one is the size of the molecule. Smaller molecules like oxygen and carbon dioxide cross membranes more easily than larger macromolecules, which face greater resistance. Next is the polarity. Non-polar lipid-soluble molecules diffuse readily through the lipid bilayer, while the polar molecules require specific transport mechanisms. Third one is the charge. Charged particles cannot pass freely through the hydrophobic core and therefore depend on ion channels or pumps for transport. The fourth one is the concentration gradient. A steeper gradient enhances diffusion as molecules naturally move from the regions of higher concentration to lower concentration. Next, we will discuss the overview of transport mechanisms. In this, the first one is the passive transport. Passive transport moves molecules down the concentration gradients without energy, enabling diffusion of small non-polar molecules and facilitated transport via ion channels and polar substances. Next is the active transport. Active transport powered by ATP or ion gradients moves substances against gradients, exemplified by sodium or potassium ATPase, crucial for membrane potential and secondary transport. Third are the proton pumps. Proton pumps like sodium-potassium and calcium-calmodulin systems regulate pH and ion balance, vital for signaling and bioenergetics. Passive transport. First, we will see the features of passive transport. Passive transport is the movement of molecules without energy expenditure. It is driven by concentration gradient or electrochemical gradient. It always moves molecule from high concentration to low concentration. Next are the types of passive transport. The first one is the simple diffusion. Non-polar hydrophobic lipid-soluble substances such as oxygen, carbon dioxide, nitric oxide, alcohol and steroids diffuse through the phospholipid regions of plasma membrane. No carrier proteins are required and its rate depends on concentration gradients and lipid solubility. Next are the factors affecting the rate of simple diffusion. The first factor is the temperature. Diffusion depends on particle kinetic energy which raises with temperature. Warmer conditions increase molecular motion, so diffusion is faster. For example, sugar dissolves more quickly in hot tea than cold. The second is the molecular weight. Smaller, lighter molecules like gases diffuse faster while heavier ones such as proteins move slowly. Third one is the steepness of concentration gradient. A greater difference between two regions increases the diffusion rate as particle moves rapidly down the gradient. The fourth one is the membrane surface area. Larger areas like microvilli in intestinal cells provide more sites for diffusion, enhancing transport efficiency. The fifth one is the membrane permeability. The rate also depends on how permeable the membrane is to specific molecules. For example, potassium diffuses faster than sodium. Now we will discuss about facilitated diffusion. Facilitated diffusion involves carrier proteins that transport molecules passively across the plasma membrane by binding on one side and releasing unchanged on other side. A solute attaches to a carrier, causing a conformational change that moves it across carrier functions for both cellular membranes and organelles. This process is rapid one. Carrier can move about 1000 glucose molecules per second. Like ion channels, carriers move substances down their concentration gradient without energy input, confirming it as passive transport. Carriers display saturation. Transport rate rises with solute concentration only up to a maximum. Once fully occupied, carriers cannot increase transport further as no additional capacity remains. The third one is osmosis. Osmosis is the passive diffusion of water across semi-permeable membrane, moving from hypotonic, that is the low solute, to a hypertonic, which is a high solute solution driven by osmotic pressure to achieve equilibrium, requiring no energy. It is governed by osmotic pressure with water moving from the areas of low solute concentration, which is the hypotonic, to high solute concentration, which is hypertonic, until equilibrium is achieved. Specialized channel proteins called as the aquaporins dedicated to water transport facilitate water transport significantly, enhancing osmosis compared to diffusion through the phospholipid bilayer alone. Cells can dynamically control osmosis by inserting or removing aquaporins, critical for maintaining cell volume and responding to environmental changes, thereby increasing or decreasing water permeability. In kidneys, aquaporins regulate water reabsorption, preventing excessive urinary loss. For example, 180 liters per day without regulation, crucial for fluid homeostasis. Dysregulated osmosis due to aquaporin defects contributes to conditions like cerebral edema and cancer cell migration. Kidney cells finely regulate aquaporin activity to control urinary water loss. Without this regulation, the human body would excrete 180 liters of urines daily, requiring equal water intake. Unlike ion channels that move charged solutes, osmosis specifically concerns water transport across membranes. Now we will see osmolarity and tonicity. First, the osmolarity. It refers to the total concentration of solute particles, that is the permeating and the non-permeating, in a solution expressed in milliosmoles per liter. It is a quantitative measure. It is same for both permeable and non-permeable solutes. For example, for plasma, it is around 300 milliosmoles per liter and does not directly predict the cell volume change since permeable solutes may not affect water movement. Next is the tonicity. It refers specifically to the effect of non-permeating solutes on a cell volume and pressure. A functional measure describing cellular response such as swelling, shrinking or no change depending on whether a solution is hypotonic or hypertonic or isotonic. It directly determines water flow and thus cell fate. Hypotonic causes swelling, hypertonic causes shrinking, and isotonic maintains normal shape. Now we will see the biological significance of passive transport. It provides oxygen and nutrient supply. It helps in removal of carbon dioxide and metabolic waste. It regulates cell volume and osmotic balance. It helps in maintenance of resting membrane potential. Dear students, we will now discuss about active transport. Active transport is the energy-dependent movement of molecules across membranes, usually against their concentration or electrochemical gradients. It requires ATP hydrolysis or ion gradients. This process is essential for maintaining concentration differences of ions and other substances across the cell membrane, which are critical for various cellular functions. It ensures cells can accumulate essential nutrients, maintain ion balance, critical for processes like nerve impulses and muscle contraction, and expel the waste even under unfavorable conditions. There are two main types of active transport. First one is the primary active transport, ATP as the energy source. Primary active transport directly uses ATP to fuel the transport of molecules. It directly utilizes ATP hydrolysis to drive molecules against gradients. The sodium-potassium pump, discovered by Jens Koh, who won the Nobel Prize in 1997, pumps 3 sodium out and 2 potassium in per cycle, consuming 25% of neuronal ATP to sustain membrane potential, which is around -70 millivolt. Examples include beyond sodium-potassium ATPase, calcium ATPase, and hydrogen ATPase pumps regulate intracellular calcium and pH respectively, critical for signaling and bioenergetics, with PG research targeting their dysregulation in diseases. Now we will see the secondary active transport, harnessing ion gradients. Secondary active transport, also known as co-transport. Secondary active transport leverages ion gradients, for example, sodium from primary transport to co-transport molecules. The sodium-glucose linked transporter, or the SGLT1 in intestines, absorbs glucose. The first one is the symport or the co-transport. It involves the simultaneous movements of 2 molecules in the same direction across the membrane. An example is the glucose-sodium symporter, which uses sodium gradient created by sodium-potassium pump to transport glucose into the cell. Second one is the antiport or the counter-transport. Antiport, on the other hand, involves the movement of 2 molecules in opposite directions. Example is the sodium co-transport of glucose. The third one is the vesicular transport, moving the large cargo. It is an ATP-dependent mechanism for transporting macromolecules via membrane vesicles through endocytosis and exocytosis. Endocytosis, which means engulfing substances into vesicles. Endocytosis includes phagocytosis, which is the uptake of large particles, pinocytosis, which is the uptake of fluid, and the receptor-mediated, which includes clathrin-coated pits internalizing LDL cholesterol. Second one is the exocytosis, which means the secretion of neurotransmitters and hormones and proteins. Vesicular transport defects are linked to diseases like Alzheimer's, which is the amyloid clearance impairment. Dear students, let us now have a look at the biological roles of active transport. It maintains iron gradients essential for nerve impulses and muscle contraction. It regulates intracellular calcium for signaling. It enables nutrient absorption in the intestine and kidneys, and it preserves cell pH, osmotic balance, and homeostasis. It also provides therapeutic targets. For example, sodium-potassium ATPase inhibitors like ouabain in hypertension and cancer research. We will now see the proton pumps and the ion transport systems. Proton pumps are membrane proteins that actively transport hydrogen ions across membranes using ATP. They establish a proton gradient or the proton motive force that powers vital processes like ATP synthesis in mitochondria, chloroplasts, and bacteria. First are the types of proton pumps. The first type is the F-type ATPase or the ATP synthesis. It uses proton gradients to synthesize ATP during oxidative phosphorylation and photosynthesis. The second one are the V-type ATPases. It pumps protons into vesicles and organelles like lysosomes or vacuoles, ensuring acidification for digestion and signaling. The third one are the P-type ATPases. These are ATP-driven proton pumps, for example, plasma membrane hydrogen ATPases in plants and fungi that regulate pH and nutrient uptake. Next are the ion-specific transporters. In this, the first one is the sodium-potassium ATPase pump. The sodium-potassium ATPase, a primary active transporter, maintains electrochemical gradient by hydrolyzing ATP, crucial for nerve impulses and secondary transport. Dysregulation linked to heart failure targeted by digitalis glycosides. Next are the proton pumps. Proton pumps or the V-type hydrogen ATPase acidify endosomes and lysosomes essential for degradation and signaling. In cancer, V-ATPase overexpression promotes metastasis. In cancer, their overexpression promotes metastasis, and inhibitors, for example, bifilomycin, are studied as potential therapies. Next are the calcium-calmodulin systems. The calcium-calmodulin systems regulate transport via calcium binding to calmodulin, activating channels like IP3 receptors for signal transduction. Dysregulation contributes to cardiac hypertrophy and neurological disorders. The biological importance of proton pumps. First one is the energy metabolism, which is crucial for oxidative phosphorylation and photophosphorylation. Intracellular pH regulation, which maintains cytoplasmic pH and organelle acidification. Next is the vesicular transport, which enables endocytosis and exocytosis via endosome-lysosome acidification. Next is the bone remodeling, in which osteoclast proton pumps dissolve minerals for bone resorption. Next is the pathogen survival. H. pylori proton pumps resist gastric acidity. We will now see the sodium-potassium pump or the sodium-potassium ATPase. The sodium-potassium pump is an integral membrane protein found nearly in all animal cells. It was discovered in 1957 by Jens Christian Skow, who received the 1997 Nobel Prize for this breakthrough. This pump is essential for iron homeostasis, particularly in excitable cells like neurons, which rely on it to transmit impulses. First one is the structural organization. The alpha subunit, which consists of the catalytic core, binds ATP and sodium and potassium. Next is the beta subunit, which have a regulatory role and stabilizes the complex. The third one is the gamma subunit, which modulates pump kinetics in certain tissues. We will now discuss mechanism of action. It uses ATP hydrolysis to pump 3 sodiums out and 2 potassium in per cycle. It generates a negative resting membrane potential, which is around 70 millivolt, and prepares nerve and muscle cells for action potential transmission. The biological function includes that it restores ion gradients after action potential, provides energy for sodium-linked glucose and amino acid symport. It maintains electrolyte balance, pH and waste filtration. The isoforms regulate motility and acrosome activity. The astrocytes and the neurons use massive ATP, which is around 75% in grey matter, to fuel sodium-potassium ATPase, ensuring neurotransmitter reuptake. The overexpression of pumps in hyperthyroidism raises basal metabolic rate, oxygen consumption and calorigenesis. Dear students, let us now see what is its clinical relevance. The first one is the cardiac therapy. Target of cardiac glycosides like ouabain, digoxin, which increase heart contractility in heart failure. Second is the hypertension and neurological disorders, which are linked to pump dysfunction. The third is the fertility and metabolic disorders. The isoform defects affect sperm function and systemic energy regulation. Let us now have a look at calcium pumps and calmodulins. Calcium transport mechanisms. Intracellular calcium kept very low, which is around 100 millimoles, compared to extracellular, which is around 1.8 millimoles. This steep gradient allows cells to use calcium as a rapid and sensitive signaling messenger, and calcium influx or efflux, tightly regulated for signaling, entry and removal of calcium, are precisely controlled to ensure that signals are transient and do not disrupt cellular homeostasis. Calcium ATPase pumps. Plasma membrane calcium ATPase, or the PMCA, actively pumps calcium ions out of the cytosol into the extracellular space to restore resting calcium levels after signaling events. The sarcoplasmic endoplasmic reticulum calcium ATPase, or SERCA, it transports calcium from the cytosol into the endoplasmic reticulum, or SR, where it's stored for future release during the signaling or contraction. It maintains low cytosolic calcium. These pumps keep intracellular calcium at nanomolar levels, preventing toxic accumulation and ensuring cells remain responsive to new stimuli. Sodium calcium exchanger. The first one is the secondary active transporter, which uses the energy stored in sodium gradient to move calcium against its concentration gradient. Second is that it exchanges 3 sodium ions for 1 calcium ion. This exchange efficiently removes calcium from the cytosol, especially during rapid changes. The third is that it is critical in cardiac muscle relaxation. By expelling calcium after contraction, it helps reset the muscle fibers for the next heartbeat. Next are the calmodulins, the calcium sensors. It is a small calcium binding protein which is around 17 kilodaltons and highly conserved proteins found in all eukaryotic cells. It binds up to 4 calcium ions via EF hand motifs. Each binding induces conformational changes, allowing calmodulin to regulate diverse target proteins. The calcium calmodulin complex activates many enzymes like CAM-dependent kinases, phosphatases, and nitric oxide synthase, linking calcium signals to cellular responses. Now, the role of calcium and calmodulin in cells. First of all, it helps in signal transduction. It converts brief calcium spikes into sustained biochemical cascades, amplifying signals. The muscle contraction, which activates myosin light chain kinase, which regulates actin-myosin interaction. Next, the metabolism. It stimulates enzymes such as phosphorylase kinase to enhance glycogen breakdown and energy release. It also helps in gene expression, where it regulates transcription factors like CREB, influencing long-term cellular adaptation. It also helps in neurotransmitter release like calcium influx at nerve terminal triggers vesicle fusion, enabling synaptic communication. Let us now have a look at the clinical aspects. Abnormal calcium signaling linked to arrhythmias, Alzheimer's, cancers and dysregulation leads to disrupted excitability, neurodegeneration and uncontrolled proliferation. Calmodulin mutations cause calmodulinopathies affecting cardiac rhythm. These rare genetic disorders impair calcium sensing, predisposing patients to life-threatening arrhythmias. Dear students, let's have a look at experimental approaches in membrane transport studies. The first one is the electrophysiology, in which patch clamp technique is used to study the ion channel activity. The second technique is the imaging technique, in which fluorescent dyes like Fura-2 for calcium are used, and the confocal and super resolution microscopy are used for transport dynamics. The third are the molecular biology tools, in which gene knockout and knock-in is used for transporter, CRISPR-Cas9 to edit pump channel genes. Next are the biochemical approaches, where ATPase activity assay are used in radioisotope tracer to study the ion flux. Dear students, this course has shown us how passive and active transport proton pumps like sodium-potassium ATPase, calcium pumps and calmodulin orchestrate the rhythms of cellular physiology. As we conclude, remember that understanding membrane permeability and transport is not just about memorizing mechanisms, but about seeing how the movement of ions and molecules sustain the spark of life itself. Carry this knowledge forward with curiosity and confidence, for it will guide you in both innovative research and unraveling the complexities of health challenges like cancer and neurodegeneration diseases. Dear students, this was all about today's lecture. I will see you next time. Thank you.